Cells require energy to drive certain processes in the cell. They have to extract this energy from two main processes, either cellular respiration or fermentation. Cellular respiration is the breaking down of glucose into ATP and other byproducts such as carbon dioxide and water. There are two types of cellular respiration, aerobic and anaerobic. In order to generate ATP, this process uses either substrate level phosphorylation or oxidative phosphorylation. There are 4 steps of cellular respiration: glycolysis, an intermediate step, the Krebs cycle, and an electron transport chain. Step 1 is glycolysis. Glycolysis is the initial breaking down of glucose. This step requires glucose and an initial investment of 2 ATP. To understand, you can think of this initial investment like gambling. In order to win money, you have to put in some money. So essentially, this is what we are doing here. In order to gain ATP out of this process, we have to invest 2 ATPs. The basis of glycolysis is that through phosphorylation, one glucose molecule is essentially split up into 2 pyruvate acid molecules. During the intermediate step, the 2 pyruvate molecules that were made in glycolysis are oxidized, forming a 2-carbon compound, acetyl coenzyme A. Since pyruvate was a 3-carbon molecule and acetyl is now a 2-carbon molecule, this means one of those carbons from each pyruvate molecules is now given off as carbon dioxide byproduct. During this step, acetyl coenzyme A is completely broken down, which will end up producing ATP and reduced cofactors NADH and FADH2. These cofactors have high energy electrons that are then carried into the electron transport chain. After the first 3 steps, we now have 4 ATPs, 10 NADHs, and 2 FADH2s. The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons are being passed from one electron carrier to another, and while this is happening, protons are being pumped across the membrane. When enough protons have been accumulated, they then flow through the membrane via ATP synthase to synthesize ATP through substrate-level phosphorylation. After this step, we now have 34 ATPs, which brings us to a maximum total of 38 ATPs at the end of this process. Because some organisms lack a final electron acceptor or a respiratory chain, these organisms rely on fermentation. The main goal of fermentation is to sustain ATP production rather than make it. In fermentation, the final electron acceptor is now the pyruvic acid from glycolysis. In fermentation, only 2 to 3 ATPs are produced, which makes this process a lot less effective.